Morning. All right. So I'm going to talk about systems over today, this morning, and tomorrow morning. I wanted to talk about systems which can be found in more than one state, or multiple states, and two states in particular. And I wanted to give you some very basic tools from dynamical systems theory to try and understand these kind of what we'll call bistable states. So bistability is a notion that I will spend some time on and tell you how someone like me who's a theoretical physicist, thinks of it mathematically. And I'll give you some examples from, I'll introduce the idea of a cell, say, having two states or being capable of being in two states, using the example of temperate bacteriophage. I'll come to that in a second. But also tomorrow, with Sunil, we will look at some of the consequences of other kinds of cells, like yeast being in two different states, and see at the population level what can happen if you have cells that have two different states. So that's tomorrow. Today, I will stick with temperate bacteriophage, but I should mention that I hope a lot of the tools and other things I talk about today and the notions of bistability will be of use to you in several subsequent talks. For instance, in fact, even the next talk which Sunil will give, where he'll talk about heterogeneity in cellular populations, that's also something that requires and uses the notion of multiple states that cells can be in. And I hope in Andrash's talk on cell differentiation, you will also see places where you can use some of the notions I will talk about when thinking about cells that differentiate into multiple different states, or at least have the potential to do so. Also, I would like you to, make connections back with, say, the talk Vidya gave or Stuart Newman gave, and think about developmental situations, and think about cells taking on multiple states there. And perhaps some of the ideas and notions we talk about during this lecture will help you make connections over there and help you understand how perhaps one can think about them sometimes a little more quantitatively or mathematically, and whether and when that would be of use in understanding these systems. Okay, so that's just to give you a sense of what I want you to take away from all this. How many people know what bacteriophage are? All right, some do, some don't. Bacteriophage, for those who don't, are little organisms that they're viruses that infect bacteria. And there are many different types, of course. And I will not talk about them in any generality, but I will talk about specifically temperate bacteriophage, and I'll describe them to you in a bit. And in particular, one very famous example of temperate bacteriophage, which is phage lambda. Those of you who know a little of the history of molecular biology would know that this is an organism that's been studied right from almost the beginnings of molecular biology. It played a great role in developing many of the ideas, tools at the beginnings of molecular biology. And in some ways, it's a rather simple system. In other ways, you will see, I find that even after 60, 70 years of intense study of an organism like this, I think there are two things to learn. So it's a virus that consists of a number of different proteins that form some kind of structure, a sort of a tail and a head, and the head contains DNA. And the DNA of this phage encodes, has all the genes that encode for all the proteins and other things. Proteins to regulate its behavior inside of, once it infects a bacterial cell and so on. So the interesting thing about phage lambda and other temperate bacteriophage is that they actually, when they infect a bacterium, what they do is that the DNA inside them is injected into the bacterium. And I'm going to talk about this class of phage that has DNA inside it and injects the DNA in, and nothing else. Nothing else goes in. You can think of this as a bacterial cell, and the only thing that's come in is this little small genome, and everything else has to proceed from there. Temperate bacteriophage are interesting because they can actually proceed along two very different, one can say, life strategies. They can either, so once this genome goes in, it starts using the machinery of the bacterial cell to express its genes. And those genes are expressed, they do various things. Amongst the kinds of things they can do if it goes into this pathway is replicate that genome. So you can start having multiple copies of that phage genome now after a while. And you can start then expressing other genes that produce all these other proteins I told you about, the things that would form the structure of the phage. And you can start seeing that little particles that look like the phage start forming. And eventually, the cell lyses and bursts, and the bacterial cell is dead then, but it releases a large burst of offspring phage. So essentially, this is a replication strategy where the bacteriophage uses the machinery of the bacterial cell to produce a lot of offspring, usually on the order of about 100 per bacterium that's infected. Okay? So that's one life strategy. The other thing that can happen is that it can actually go dormant. And the phage genome can get integrated into the genome of the bacterium and sort of sit there happily ever after, more or less. It can sit there until something signals it to jump out and jump back into that phage. It can sort of, sometimes genes that the phage uses, some genes can still be active in this role. Amongst the kinds of genes that can be active are genes that suppress that pathway, that prevent it from replicating, prevent it from jumping out of the genome. Sometimes you can even have genes that actually help the bacterium. There are a few cases where there are genes that the phage encodes, which helps a bacterium, say, metabolize something a little more efficiently or protect against antibiotics. So sometimes there can even be an advantage, but very often there's no particular advantage for the bacterium other than that it doesn't die. So these are two very different sort of life strategies, let's say. And this is a decision, whether to go into this strategy or that strategy, is a decision that's made sometime a little after the infection. And that's one of the questions that's, to some extent, still being studied is, when does that decision really get made? When is it settled? How long does it take? But typically, it would take a few minutes, at least. What else do I want to tell you about this? Essentially, there are aspects to this decision that are deterministic and aspects that are stochastic. By stochastic, I mean dependent on absolute chance. You know that there are many processes that happen in the process of gene expression or protein production that will be subject to a certain amount of chance. Some particular protein might just get produced, or a certain gene might just get expressed a little bit more for a short period of time, or a little later or a little earlier by pure chance. These things can affect whether this decision happens and goes this way or this way, depending on how early that happens. But there are also very deterministic aspects to it in the sense that various kinds of signals can bias whether you go that way or this way. It can bias the probability, even if it is a stochastic process. So we know, for instance, that under certain conditions where there are lots and lots of viruses and very few bacteria, then you tend to go into the dormant stage, into the dormant pathway more often. We know that if the cell is a little bit closer to stationary phase, it's starving, it's not doing very well, you tend to go more into the dormant phase. There are things like that, and there are other things that would push it towards that way. So that's something to keep in mind, that there are deterministic and stochastic aspects to it. So why do people think of this in terms of bistability? You can sort of think of this state as being potentially a stable state. Like I said, it does, once it goes into this dormant state, it does stay there for a while, unless a certain specific signals come in that make it jump out and jump to that state. It's actually quite stable. But over here, you can see that this is really not a stable state in any sense, right? If you go into this pathway, eventually the cell is going to burst and die. So that's not particularly stable. It is a different sort of state, but it's hardly stable in that sense. But nevertheless, people talk of this kind of system as having an underlying bistability, an underlying setup such that this phage inside a bacterium can actually be in two stable states. And the reason they talk like this is because people have done experiments where they've prevented the lysis. They let most of this process happen, but they block lysis. They block the proteins that cause lysis. In that kind of state, you can you can actually find that the system settles down to a particular state. And that state is quite stable. And people have done this at various stages. You can even stop it a little earlier in replication. You can prevent too much replication of the bacteriophage DNA. And then also it sort of settles down into a state that seems fairly stable. And by stable, I mean, what is the chance that it sort of jumps out of the state and moves into the other state or some other state per cell generation? And the numbers that people have measured are pretty small. So in that sense, there are two states, and people often call this the immune state and the anti-immune state for a different reason. We don't need to go into that. But there are two states, and they are both intrinsically fairly stable. If you, for now, forget about the idea that actually, really, in one state, it eventually kills the cell, and that whole setup is gone. But if you block that process, you could actually produce two rather stable states. And if you looked at, yeah? So that's partly why it's called the immune state. Bacteriophage can attack it. So the question was, in the immune state, can another bacteriophage come, infect, and then what will happen? And it so happens that in the case of lambda, at least, and a whole class of such bacteriophage, this state is immune to further infection by the same phage or related phage, a certain class of related phage. And it's immune in the sense that the phage will come, it will insert its DNA, but nothing will happen. That DNA will just sit around. It won't express very much. And the reason is rather simple. It's because there is a protein, a phage protein being expressed in this state that is suppressing all this, all the genes that do all the other stuff. So when a new phage comes in, that same protein which is floating around in the cytoplasm also suppresses the new genome. That is why it's called the immune state. And that, for, I suppose, just to be contradictory, is called the anti-immune state. So there is some sense in which this system is capable of two states, and each can be pretty stable. Right? Everyone with me so far? I should also mention that, maybe I didn't say this very clearly in the beginning. The replication of the genome happens to some extent in whichever pathway you go. So if you go in this pathway, obviously you replicate and you produce many copies, and eventually you produce, say, 100 copies. But there's also replication in the beginning when going into this state, into this pathway. There are 2 or 3 rounds of replication before the decision is made that things will go dormant. So actually, maybe a better representation of this state would be initially there would be 2 or 3 genomes over here. One of the genomes gets integrated, and the others are just floating around. And eventually, as the bacterial cell divides and subdivides and divides, these get diluted out. So I should point out that the stability of this state is seen at various numbers of genomes. This state is a very different state than this state, and it's stable. And that can be true even if this has 1 genome and this has 1 genome, or this has like 8 genomes and this has 8 genomes. So sort of irrespective of the number of genomes that are there at any given time, you can have 2 states. We'll come back to this later in the talk, but just keep that somewhere in the back of your head. Okay, so this is a case. I mean, maybe I should try to point out over here a sort of an open question for you to think about. To what extent is this a useful analogy or model system for a developmental kind of decision? You are very familiar, perhaps, from previous talks with states where a cell differentiates into one state, and the same cell is capable of differentiating into a completely different state. And some signals, plus stochasticity, as we might see in Andras' talk, are driving these decisions of whether the cell state goes into this state or into the other state. Does it become a muscle cell or a neuron cell? To what extent is this kind of a decision that's happening in a completely different organism, and perhaps in a simpler setting, to what kind of extent can one say that this is a developmental decision? I think, in fact, that there are many things to be learned about developmental decisions from simple decisions like this. But this is an open question. It's just something we could discuss at some point. Okay, so I want to proceed by sort of telling you that lambda phage in particular has been really well studied. Its genome is completely sequenced. We know quite a lot about almost every gene that's in there. We know that there are various genes that are used in the early lytic part. That's the stage where you're replicating the genomes. There are things that are late lytic. So the head is the set of genes that produces the proteins that forms this structure into which the DNA is held. But the most important part of the regulation of all this and these decisions is also reasonably well known. And two proteins are particularly important, C1 and CRO. And there is a model that people have had for a very long time of what regulation underlies this bistability, this fact that temperate phage can go into two very different states and be stable in either of them. And this is the standard model. So the model is that, and you can read about this this is almost like textbooks. It's gone into the textbooks. This is the way people think lambda phages, multiple stable states are formed. There is a protein called C1 and a protein called CRO, and each inhibit the other's transcription. So this is a rather simple motif where you have two proteins, and each is inhibiting each other. And you can see, just as a hand-waving argument, that this could be in two stable states. You could have a state where there's lots of C1, for some reason, lots of C1 and very little CRO. And in that state, C1 being high would be inhibiting further production of CRO. And that's it. Since there's not much CRO, this inhibition doesn't work and C1 stays high. Or you could have the other way. It's sort of, here it's all symmetric, where CRO is very high and it's inhibiting C1 and C1 is very low. So one can sort of see that this motif could produce two completely different states. And you could imagine things being stable in those two states, so far in a purely hand-waving way. And people have indeed observed that is the case. So they've measured C1 in this state, CRO in that state, and that is indeed true. So in a sense, these two proteins are markers of which state you're in. And it's fairly certain that they are playing very important roles in setting up this decision. Okay? However, things are always much more complicated than that. If you really start looking at which proteins and which genes are interacting with each other, it rapidly gets more and more complex. So here you can see C1 and CRO. This was the little motif I was talking about. C1 inhibits CRO, CRO inhibits C1. Don't worry too much about the details here. The point of this slide is just to show you that there's quite a lot going on. There are many other genes. There's something called C2 and N. Lots of things are interacting with lots of things. And all of these are involved in the decisions. And here, this picture tries to give you a representation of also how external signals like poor nutrition could affect the phage genome and affect gene expression at the phage level, and therefore bias this decision. I said that there was both deterministic and stochastic aspects to it. Some of the deterministic aspects are these external signals that come in and affect various things over here. So some of the complexity of this regulatory network might simply be because it has to deal with all these signals and make sure that, oh, if that signal comes in, I better go more often into the lytic pathway, or if that signal comes in, I better go into the lysogenic pathway, and so on. So some of the complexity could be that. And it could still be true that this little motif is what underlies the possibility of two states in the first place. But it's not so obvious looking at this. It could be that, oh, maybe C2 is very important for even the existence of two states. And so these are the kinds of questions one would like to investigate. What are the different roles that all these things are playing in the decision? What are the ones that are setting up the two states in the first place? And what are the things that are biasing or regulating and fine-tuning which one you'll go into? Okay? So people, as you may have heard in various discussions, I know there have been discussions of top-down approaches, bottom-up approaches. People can study this in many different ways. A top-down approach would take as much information we have and start from there, perhaps build a model, try to then start tweaking things and ask what changes, what happens, is this absolutely necessary for the two states or not? A bottom-up approach might start from this simple thing and then start adding little pieces. So what we're going to do in the rest of this lecture is do more of the bottom-up kind of approach. What I want to do is maybe if you were take the question to be, what if lambda phage was just this motif? In that case, can we understand why, when you would get two stable states? Of course, naturally it can't be true all the time. This motif, of course, from a logical argument, seems to produce two stable states, but that surely must depend on various parameters. There are lots of parameters to think about, right? What about the stability of these proteins, their half-lives? What about the rates of production? Can you still get two states if C1 is produced a lot, but CRO has a low level of production? Will it still work? Do they have to be symmetric? We can ask many questions, right? We can ask, do these strengths of these interactions have to be about the same? Or can one be very much stronger than the other and it will still work? So these are the kinds of questions one can ask if you start with a very simple motif. So that's what we're going to do. So what I plan to do now is maybe over the next 40 minutes or so, 45 minutes, use just the board. We'll talk a little about bistable systems. We'll go over certain tools that you can use from dynamical systems theory to understand very simple motifs of bistable systems. And then maybe at the end, if there's time, I'll come back to the slides to show you some more research questions. And just to give you a flavor of what we can use using the tools that I have presented, can one go back and ask interesting questions about, say, lambda phage? Okay, so what I want to do now is, let's stay with this motif. So please tell me if I'm, if I start writing too small and nobody can see it or anything like that. Everybody can see this size, right? So if you want two states and bistability, the notion has two parts to it. One is that you want at least two steady states. Right? And I will define to you what I mean by a steady state in a minute. And you want the steady state, and you want two of those, if we're talking about bistability, two of these to be stable. And we need to define stability also. So both are notions that we need to define. And what I want to do is to write down some mathematical models of a simple motif like this, and ask whether, using those mathematical models, can you show both these properties? And if so, what does it tell you about the parameters? What parameters will work? What won't work? What will give both properties? What will not? And one of the points of working from bottom-up and very simple motifs is to try and be as general as possible. So I will try to keep most of the lessons we learn as general as possible and try not to make too many assumptions, but that's almost impossible. You always have to make some assumptions. So to begin with, let me write down one mathematical model which is specific, it makes certain specific assumptions. You can keep that as a particular example in the back of your mind in all the subsequent analysis. And we can talk about what properties that I tell you are specific to that particular model and what are more general. So let me write down one kind of model which is actually quite popularly used in trying to understand this kind of motif. So I'm going to put down, just to make sure my notation is consistent, I will keep track of my notes. So let me just write it down, and some of you will be familiar with these kind of equations and some of you won't, I'm aware of that. So we'll go over this in some care. So what's happening here is on the left-hand side, I am trying to write down the rate of change of the concentration of these two proteins. And I've almost got it. So how many people are familiar with these kind of equations? On the left-hand side, for those of you who are not, this quantity C1 crow, wherever it appears, represents the concentration or number. It doesn't really matter at the level I'm going to discuss it. It represents, say, the concentration of C1 in the infected cell and the concentration of crow. And this represents the rate of change of that concentration with time. So these are equations that tell you that if you know what crow and C1 were at any given time, let's say at time 0, then you could use these equations to calculate what they will be at the next time step, let's say a little bit, like a second later, and use that to calculate what will happen a little bit later. And so you can get the full time course of how these concentrations will change with time. Okay? If you have equations like this. Alpha, K, H, gamma, all of these are parameters. And we need to talk about what these parameters mean. And why do I have? You can already see, even in a very simple model, I have so many parameters already. But let's think a little bit about, as we think about the parameters and so on, what these equations mean and what kind of assumptions go in. So already, without knowing anything further, you already should see that I've made one very big assumption. And that assumption is I'm representing everything about C1 and crow and these two proteins by a single number that changes with time. So I have no concept of space. I am not talking about the concentration being different in different parts of the cell. I'm talking about a single concentration for the whole cell. So I've already made a big assumption that the spatial distribution of these proteins inside the cell does not matter. Right? Maybe we should write down some of these assumptions. I'll put them on the side. So one assumption is the system is well mixed. It's just another way of saying what I said. There are no spatial components. Everything can access everything. And I'm going to represent the whole system by simply one number for each protein. That number will, of course, change with time. One can do much more complex things. I think Vijay, in his lecture, will talk a little more about situations, especially pattern formation in space, where you can't ignore space, and where the concentrations of morphogens, for instance, and their spatial localization is very important. Then you need to keep track not just of the protein concentration all over, or one number that represents it, but you need to know at each point in space what the protein concentration is. That's a different kind of model and a different kind of representation. But here we'll stick with this well-mixed stuff. Okay, what about terms like this? Some of you are familiar with terms like this, right? Every equation here has two types of terms, one a positive term and one a negative term, which really should represent all the processes that produce C1 are somehow represented by the positive term. All the processes that degrade C1 or reduce its concentration are represented there. Now, I've made some assumptions here already. I have written a very simple form for the degradation. So, can anybody tell me what assumptions I'm making here? What kind of degradation process can a term like this represent? Yes, so somebody said that the rate of degradation is constant. So indeed, gamma is something like a rate of degradation. So if for those of you who have looked at, for instance, radioactive decay and so on, that is an analogy to this. Suppose you have a protein and it just has a certain half-life. You can think of it very similar to the way you think of radioactive decay. There is a certain half-life. And if there's nothing else that's changing this half-life along the time course of all this, and the half-life is a constant, then you can think of every molecule has a certain probability per unit time to decay and just degrade. And that's the kind of term that we've written down here. In the case of, so one can ask now, is this a sensible assumption to make? So we've taken, we've taken linear degradation. Very often in cells, that may be a pretty bad assumption. You know, some of you must know, that in cells there are often proteases. And proteases are proteins that will actively go and degrade other proteins, say, for instance. And these proteases, their concentrations themselves may change up and down. In that case, in a situation like that, this would be a very bad assumption. Okay? But we'll go with this assumption. In the case of C1 and CRO, it doesn't seem to be too bad an assumption. People have actually measured, if you stop the production of C1 and CRO and tried to measure using various experimental techniques, don't ask me the details of them, but looked at what happens to the concentration. It goes down exponentially. And this kind of term gives you that. If you want to know more about it, we can talk about that later, but let's go with that. But at least, you should be aware of the kinds of assumptions, and you should always be questioning what kinds of assumptions is gone. What about these kind of terms? Has anybody seen these kinds of terms before? Do they look familiar? Yeah? One or two people have. Let's see, to begin with, whether this at least has some features that we want. We were talking about C1 being repressed by CRO. And by repression, I meant transcriptional repression. And in turn, CRO being repressed by C1. So if you're thinking about transcriptional repression, is this a sensible term to use or not? So one way one can think about that is to just say, well, does it have the right features? What do you want from transcriptional repression? If you want CRO to be repressing C1, which I will write like this, what do you want? You want the rate of production of CRO, the rate of production of C1 to, as a function of the CRO, to start sort of high when there is very little CRO and to come down, right? That's more or less what you mean by inhibition or repression. So does this have that property? That's the first thing to check. And it does. It has something like, it looks a bit like 1 over 1 plus x to some power, like let's say x squared if we had chosen x squared. Sure, that's true. As x increases, you would start somewhere and go down. No problem. So it at least has that property. As you change these parameters, you can see that various kinds of shapes can emerge. So suppose h was equal to 1, the shape would actually look like this. Now we're just plotting. We're not even really thinking about mechanisms and so on. We're just saying, what kinds of shapes can you get? You could get a shape like this. You can check this out. Please try to plot it yourself in something like MATLAB or something. If h increases, and let's say is larger than 1, let's say it's 4 or something, you can see that a little plateau-like thing can arise, and it can go down. You can get shapes like that. If you change the k and increase that, then you can shift where this shape will happen. So if you increase k, you could get the same shape, same looking shape, but somewhere else in this curve. So that's just to say that there are various kinds of shapes. So one justification for looking at terms like this could simply be, it has the right properties, it's supposed to come down, it's supposed to be a repression, it does that, and using these parameters, we can explore a fair variety of shapes. You can get plateaus, you can get steep things, you can get shallow things. Oh, it's okay, it's a fair representation of the kinds of different shapes you could get. That's one way. But of course, it would be reassuring to see if you could get something like this from some kind of molecular mechanism, at least one. Then you would possibly be a little more reassured that that's okay, that's a reasonable thing to do. So let's do that too. So here we've only just shown that it covers a wide variety of shapes that you might want to explore. But let's think of an actual mechanism. So now let's think about transcription in more detail. What do we want? We want CRO to be a transcription factor that inhibits the expression of the gene that's producing C1. Well, how does that usually happen? It goes and binds somewhere, right? There's a binding site. Maybe that binding site is near the promoter region. Maybe the binding site is in the middle of the coding region. Somehow, CRO goes and binds to that binding site. And in that, when it's present in that binding site, it prevents the polymerase from going through and expressing this gene. So can we understand that binding? And can we understand whether binding and unbinding and all those processes that are involved in that could give you something like this? So I'm going to do something very similar to what Olivier did. So that's one way. You remember yesterday he was talking about proteins being in two states, either folded or unfolded. And he used Boltzmann's law to try and ask what's the probability you'll be in that state or in that state. So let's think about transcription factors and binding in a very similar way. So there are two states that you can talk about. This single binding site, for now, let's assume, there's only one binding site. It can be unbound or it can be bound, right? What's the number of ways you can have this? So if you remember, in Olivier's calculation, there were two aspects. There was the energy and there was the entropy part, which was how many ways you can realize this state. So number of ways. So how many ways? So let's be a little more specific. There is some kind of binding site over here, and there are a bunch of proteins floating around. These are all the same transcription factor. These are CRO, let's say. And let's say there are T molecules of this transcription factor. Then how many ways can you have this unbound? Somebody should give me an answer here. How many different ways are there? 2 to the power T? No, actually there's one. So the unbound state, there is only one way for it to be unbound. There are some more assumptions we're making here. We're saying that it doesn't matter. When it's unbound, we're only worried about the state of this. We're not worried where in space and so on. You know, it comes back to the assumptions we made earlier. We're not worried where are these transcription factors? Where are they floating around? If they're unbound, they're unbound. So there's only one way. But there are T ways to make the bound state, because it could be this one or that one. And those are different states. So because there are many different transcription factors, or not different in species, but many molecules of the same transcription factor, there are actually that many different ways in which you could realize this state. Another way you could think of it is if I just expanded the set of states. I said bound by molecule 1, bound by molecule 2, and so on. And then it would be one way, one way, one way. But I'm going to just bunch all of these into one state. It's not totally unlike what Olivier did. He also, if you remember, had sometimes many different ways in which you could be unfolded, but only one way in which you could be folded. It's very similar. So depending on what you're interested in, you can bunch a lot of states together as one state and just keep track of how many ways you can have it. What about the energy? So the unbound is maybe a baseline. What we know is there is an energy difference between this and this state. You will gain some energy because of the binding, correct? There's some binding energy. So this will be smaller compared to that, but the actual scale doesn't matter. The absolute numbers don't matter. So I could take this to be 0 and this to be some minus E, minus epsilon. Everyone's happy with that? Is that okay? All I'm saying here is that in the bound state, there is some energy gain. The bound state has lower energy. It's more favorable energetically. Okay, now we come to Boltzmann. So the Boltzmann weight is, as Olivier said, e to the minus the energy, so that's 0 by kBt, multiplied by the number of ways, that's just 1. And over here, it will be t e to the minus plus epsilon, because a minus sign came there, divided by kBt. This is just 1. So the Boltzmann weight is 1, and this. Everyone's okay with this? I'm really following in parallel what Olivier did last time. There's no difference. Except for him, the two states were states of proteins. For me, the state of a binding site. Is the binding site occupied or unoccupied? Maybe I should say unoccupied and occupied. And if you remember, this was the partition function. The partition function is just the sum of these. And finally, we can write down the probability of each state forming. The probability is this weight divided by the partition function. So the probability of this state is 1 plus t e to the epsilon by kbt. And this is t e to the epsilon by kbt divided by 1 plus t e epsilon by kbt. Yeah. Well, yeah, maybe I can use number of transcription factors. Would that be better? Is that less confusing? So T is the transcription factor, and NT is just the number of molecules of the transcription factor. Is that less confusing? Is that okay with you? So, what have we got? Now we know the probabilities that you can be in this state or that state. We need one more thing to try and understand what is the rate of production of your target that your transcription factor is controlling. So this binding site, these guys can bind on and off. But eventually, this is part of, let's say there is some promoter somewhere here, and there's a gene, and this gene is what is producing C1. So in the end, we want to know what's going to happen here, because we want to see if all this calculation can help us understand why we did this. So we need something that tells us the rate of production of your target in each of these states. So what is it when it's unbound, and what is it when it's bound? So let me just make a particular case. Let me say that when it's unbound, it's some alpha, some rate, some non-zero rate, and when it's bound, it's 0. That's probably not realistic in a real situation. There will probably be some small little thing, but we'll see in a second what happens. So suppose the rate is alpha and this is 0. Then it's very easy to find the net rate of production. So your net rate of production will be this value multiplied by this probability plus this value multiplied by this probability. So what do you get? You get alpha 1 plus n t e to the epsilon by kbt. Yeah? Can you see how this is very similar in form to the other one? It looks a lot like these terms with a particular value for h. H is equal to 1. So it looks a lot like alpha by 1 plus rho by k. And k is related to the binding energies, alpha is related to this alpha, and h is 1. So we have found some kind of calculation of transcription factors binding to binding sites and allowing or not allowing transcription, or allowing or not allowing protein production, gives you something quite similar to the terms we've used. So the question now is, what are the assumptions we've made in all this? Anybody want to hazard a guess on what kinds of assumptions have gone in? There's probably more than one assumption. But what assumptions can you notice? Make a guess. Okay. Every transcription factor molecule has the same probability of binding or unbinding. That's certainly one assumption we made. Is that a reasonable assumption? It's the same molecule. So you're absolutely right. It is an assumption, but I would say it's not a bad assumption. You know, let's assume these are all Cro molecules we're talking about. It's the same binding site. Should be okay that it's the same. Each individual instance of this molecule should have the same thing. Good, great, okay. Any others? Think about this whole calculation. In this whole calculation, one of the important bits we used was the Boltzmann law. Are there assumptions there in even using Boltzmann's equation? This is a tough one. If you're not already aware, you wouldn't be able to guess it. But there are a bunch of assumptions that go in here. One assumption is that everything is in equilibrium. So this whole structure and this kind of calculation works in equilibrium systems. Now, what does that mean in the context of transcription factors binding? It means that somehow, what's happening at a short time scale is things are binding, unbinding, binding, unbinding. There's lots of stochastic processes going on. But over time, it reaches some kind of equilibrium. In the classical sense of thinking about equilibrium, you can think of this as meaning that the probability of jumping from a bound state to the unbound state, or the unbound state to the bound state, has sort of settled down. It's all reached some steady state. These probabilities are not changing over time. Occupancy has, on average, reached some steady level. Okay? So there is certain assumptions of equilibrium over here. So, or equilibrium or quasi-equilibrium. So we made certain assumptions, and I'll tell you in a second what I mean by quasi-equilibrium. So that's a big assumption. Another assumption is, in all this, we assume that NT is constant. Is that reasonable? That's fine in this calculation. We can start with the calculation and assume that the number of transcription factors is constant. But in the end, we are going to take this and put it back into something like this. And here, fine, you could assume Crow is constant. But you have another equation where Crow is also changing. So now Crow is no longer constant. So is it valid, given that you have an equation where Crow is also changing? Can you really use the result of a calculation which started by saying that the transcription factor is constant? You shouldn't be able to. It looks like there is a mistake there, right? The reason you can use this to some extent is that things are happening on many different timescales. And this is a very important idea, and we use it all the time to simplify things. Suppose the changes that are happening to the Crow level from these equations are happening much slower than the binding, unbinding to this site. Then, over the time period where the binding, unbinding is happening, the rapid time period where that's happening, if that's very different from the time period over which the overall numbers are changing, you can kind of assume that it's sort of constant. So what we are saying is that if you were looking at whether, so you have time and you start with some number of transcription factors, NT, you're asking what is the fraction of time that this is bound. So if you're looking at the binding, unbinding, so let's say this is a bound state and this is the unbound state, and if I was keeping track of that, that would be happening really rapidly. Whereas this would be changing, let's say, very slowly. Maybe it goes up, maybe it goes down. But if a lot of transitions happen in any one period of time that you're looking at, you could kind of say, okay, let's assume it's more or less constant there. Lots of rapid binding, unbindings happen here, and then it changes. So during this time, is this time enough for the system to have reached equilibrium in the binding sites? If it is, then you're okay. So there is an approximation, there is a mistake you're making, but the question is that mistake and that approximation a big one or a small one? And it's a small one if timescales are very different. So this is sort of what one means by quasi-equilibrium. Of course, things are not in equilibrium. There are proteins being produced, new proteins are being produced, proteins are being degraded. Clearly, in any system like this, it is not an equilibrium system. But within that, there are many time scales. And if the binding, unbinding is happening on a very rapid time scale compared to the other processes, you could assume that part is in quasi-equilibrium. And you can use a calculation like this. Okay? So one must always somehow keep an eye on the time scales of various processes happening. What are the processes we are talking about here? Binding, unbinding, transcription, translation, degradation. So that's what we're comparing. We are comparing whether the timescales of transcription, translation, degradation, are they much slower than binding, unbinding? If this is happening on timescale of seconds, and that's happening on the timescale of hours or tens of minutes, then it's okay. Then there's a big enough gap. But if both of them are happening on the timescale of minutes, then you have a problem. Then you can't use this kind of thing. Okay? So already you can see, even in a very, very simple model setup, where we're writing really simple equations to try and describe this, lots and lots and lots of assumptions going. My main point here is not so much to prove to you that this one assumption is good or bad or whatever. We can still stick with the what-if statement and say, well, let's assume all this is okay, and still let's try and understand bistability, and we'll do that in a second. But it's important to just always keep in the back of your mind that there are lots of assumptions, and you should be aware of those assumptions, so you also know where things might break down, and where such a model is not a good idea, and where you need to change the model, or try something different, or try a different approximation. Okay, any questions about this? So far, we haven't done very much. We've taken a very simple motif, which is the kind of thing that people think underlies the bistability of phage. We've written down one possible instance of a model for this kind of motif, and we've given some idea of where these kind of terms might come from. Okay? That's it. But I think it was important to do, because it's very important to realize how many different kinds of assumptions go through. So what I want to do now, if there are no more questions about this, is I want to get back to this and actually then get back to the question we wanted to ask in the first place, is are there two steady states? Are they stable? What does that even mean? And so on. Any questions?
So why decay is not linear?
Sorry?
So why decay is not linear? Why do you make an assumption, some term one
you mean
plus?
Why this is not linear?
Yeah, why it's not
this itself, this term? Yeah. Yes. So I mean, essentially, one is sort of trying to say that this kind of term comes from transcriptional inhibition. And if you have transcriptional inhibition, the way it will depend on the concentration is not going to be linear. When the concentration is very large, you will sort of saturate to some low value of the rate of production. So if this is the rate of production, I'm just plotting that. You want something that has perhaps a sigmoidal structure. The reason for that sigmoidal structure, or if h is equal to 1, that kind of structure, is the logic comes a little bit from this binding, unbinding. When you're talking about binding and unbinding and asking what is the probability that a binding site is occupied or not, that probability is not a linear function of the concentration. And that's exactly what we saw here. Here is why it's not a linear function from this calculation. There could be, of course, certain ranges in which it is linear. For instance, you might be always working in the region where h is equal to 1, and for concentrations that are low enough that it looks pretty close to a straight line. In that case, it would be a perfectly good assumption to replace this by a linear. But you have to know it. So it would not be necessarily a bad assumption, or worse or better than this. It just depends on what situation you're in and what do you know about it. So what we will try to do in the further analysis is try and actually not necessarily be stuck to a particular functional form, and actually ask, we can certainly ask, what if this was linear? Would this still be bistable or not? And that's perfectly okay. So in the next bit, as I start analyzing this system, we can come back to questions like that and ask, will my answer about whether it's bistable or not depend on whether this is linear or not? Okay? Yeah.
There'll be some transcription factor which is causing the production of this C1, let's say. So when CRO comes, it binds to the promoter region and it inhibits.
So
is it like that?
in phage, yes, that's what happens.
So
there is a
in that
promoter
case, like when we consider particular number of molecules of CRO, we consider the probability of CRO binding is constant, right? But it will depend on the concentration of the transcription factor also.
You mean like the polymerase or some other transcription factor?
Yeah, because
who is the transcription factor in this case? So there's a situation, maybe I was a little unclear. Let's imagine you're talking about the gene which is producing C1.
Mm-hmm.
That has a promoter, and in fact, it has a binding site somewhere near the promoter where CRO binds. So there, CRO is the transcription factor. And by binding, it prevents the polymerase from binding. But in general, you're right. There could be some other transcription factor that's also involved. There could be multiple.
Oh, I thought it as a repressor. So
No, it is. It is. CRO and C1 are both repressors of each other. And they repress by binding to and near the promoter of the other one and preventing the polymerase from binding. But your question is absolutely valid in the sense of we have not used the polymerase concentration anywhere. What about that?
if it
Where
is
does that
high,
come in?
even for a given constant number of molecules
Absolutely.
of this,
So the question is, where does the polymerase come in over here? And where does the polymerase numbers come in? And where would you put in something like that? You can, of course, expand the system to also include polymerase numbers, also include that. In a sense, that's another assumption we've made. We've made the assumption in this model that all other things are constant. All other things that could affect this, ribosome numbers, polymerase numbers, all of those are constant, or in the same spirit as these timescales, to say that whatever changes are happening to those are happening much, much slower than all these changes. Okay, so those are the typical, so typically they're not constant, of course, but perhaps those changes are happening very, very slowly and under the control of something else, which is a process happening much more slower, in which case one can take this. But exactly, these are exactly the kinds of questions I think you should ask about the assumptions behind this. So we haven't even scratched perhaps the surface of all the assumptions here. We've only talked about a few. And certainly this is a very big assumption that everything else that potentially could impact this system is constant. Okay, very good. So let me come back to now, let me come back here and start doing a little more analysis of this, so we can at least get somewhere with our question of whether this system is capable of bistability or not. So what I'm going to do is now, well, I'm not sure, I can't even call this top-down or bottom-up. I'm going to take this system and actually simplify further. So this is probably a bottom-down approach, because this is already too complicated. It's in 2D, and I want to start by one dimension. So we're going to make an even simpler model where we don't have two proteins, but just one. So imagine one protein, and I will write, and I'm going to write this in a fairly general way. I'm going to keep a linear degradation term and put the rest of the production, and I'm not going to specify it. It's some function away. And we're going to look at different kinds of functions. Whatever I do here, actually I don't even need to do this splitting, but I'm just going to do the splitting just to make a few things a little easier to spot. So suppose you have this. Let's look at different kinds of functions that could be here, and different kinds of way this protein A could affect itself. So I'm going to draw the following. So on the x-axis is A, and I'm going to draw this line. This is a straight line with a slope gamma A. Okay, that's what gamma A would look like. If gamma increased, the slope would get bigger. If it decreased, it would get flatter. That's gamma A. And on the same plot, I want to plot f of A. So let's take a couple of cases. Let's take a case where A inhibits itself. So these are cases where one transcription factor is inhibiting another one, and that one is inhibiting the first one back. Now imagine a transcription factor that inhibits its own production. If that's the case, what would happen? The production, when A is 0, would be some value, right? Some level. What would happen to it as A increases? Yeah? Yes? Somebody is gesturing at least. It will go down. It will go down. Let's say it goes down like that. That's possible. Maybe there is a particular molecular mechanism that gives you a particular mathematical shape, but let's not worry about that. Anything that goes down could represent a self-inhibiting protein. What can we say from all this? First, we need to find out, are there steady states? And let me define that now. A steady state will be a particular value of A for which this is 0. Does that make sense? A steady state is simply something where the rate of change is 0. So anything where this is 0. And graphically, you can fairly easily see where that point will be. That point will be where these two things cross. So that value of A, let me call it A star, is a steady state. Why is that? The rate of production equals the rate of degradation. That's a steady state. So here, how many steady states do you have? Only one. So this certainly cannot be a bistable system because it doesn't even have two steady states. But what if there are other shapes? This may not be the shape. What if your self-inhibiting protein, the production rate depends in a more complicated way? So, looking back at the kinds of shapes we are looking at from there, well, maybe it has a plateau, maybe it's sigmoidal, it could be that. That still cuts only in one place, right? Yes? There's no way you can. What if you were allowed more plateaus? What if you did something more complicated? You could do, something like that. That one that I've drawn still only cuts it in one place. Can you imagine something more complicated that can cut it in two places? A parabola. Excellent. So that cuts it in two places. But is that a self-inhibiting protein? What are the conditions we need to put on f of A to call it inhibiting? So what does inhibition mean? It means that as you increase the concentration, the production rate should go down. Well, maybe we can relax that a bit and say, oh, okay, production rate need not always go down. Maybe it goes down some places, but it could be flat. But it certainly should not increase. Then you would not call it an inhibitor. Yes? So we're putting some constraints on FA now. We're saying, suppose it was an inhibitor, and it's an inhibitor at all concentrations. Then your f of A can at best, it can come down, it can flatten out, it can come down a bit, it could flatten out. It could do all sorts of complicated things, but it can never go up. If it can never go up, can you see that it can never cut this line in more than one place? It's just a purely geometric thing. If you have a line that's going up like this, and you have another curve that starts there and cannot increase, there is only one place. And it will cut it in one place. You can also show that, right? If it's coming down, at some point it will cross. And once it's crossed, it cannot go back up. So it cannot cross again. So we've already proven one thing. If you have a self-inhibiting protein, it cannot be bistable. Because forget about stability, it doesn't even have two states. Cannot be bistable. So the problem is with the bi part. There are no two states. So we've already got some insight. So now the obvious thing would be to ask, what would happen if you were a self-activating protein? With the self-activating protein, we'll put similar kind of condition that the production rate should go up for certain concentrations. It might plateau out a bit, but it certainly can't come down. Then you wouldn't call it an activator. Yes? So an activator has to be affecting the production rate in a way that it has to be sort of going up, maybe at least not decreasing. So now can you get more than one stable state? So remember, you can't go down and then up. You can only go up. So your parabola would still have that problem. We'll come back to the parabola later. But if I put this condition that it can only go up, can you still get two cuts? You could do something like that. So it's going up, maybe I haven't drawn this properly. It's going up everywhere, but it's going up slower and then faster. So you can have two cuts. So this is a system that potentially could be bistable, but now we have to talk about stability. So let's talk about stability. What does stability mean? Stability in this context is the notion simply that if you were at a steady state and there was some perturbation that moved it slightly away, would you come back to the steady state or would you get pushed even further away? So let's think about that in this case. Can everyone see this carefully? Where are the steady states? They are where you cut. The two lines cross. So one steady state is here and one steady state is there. And in this one-dimensional case, it's very easy to see what the stabilities of these will be. Because what you need to know is this quantity. If you move slightly away from the steady state, is this quantity going to be positive or negative? That's easy to see. If Fa is larger than gamma A, then it'll be positive. dA by dt will be positive, and A will increase. So I could represent that by an arrow. So let me find a place where Fa, so this is Fa, is larger than gamma A. This is one region there. So over here, I could draw a little arrow saying A is going to increase. And let me make that length of the arrow proportional to how much it is large. So as I go further along, the arrows will get bigger, and over here the arrows will be smaller. Does that make sense? Over here, Fa is positive, Fa is larger than gamma A, but only by a small amount. And now let me use a different color. Is this visible? Yeah, okay. I'll try and draw this thick. So somewhere over here, f of A is larger than gamma A, but only by a small amount. So let's write a small arrow there, and a bigger arrow a little further along, and an even bigger arrow a little further along. That's just representing the fact that if you are slightly on at this point, your f of production is larger than your degradation, and therefore you'll increase. Your concentration will increase. And it will increase faster and faster and faster. What about this side? This side, the production is lower than the degradation, so you will start decreasing. That means you'll put arrows this way. And the arrows will increase and become bigger until it reaches about this point. This is the point where your degradation is maybe the most different from your production. After that, it starts decreasing again, so the arrows start getting smaller and smaller and smaller and smaller. And what happens on this side? Your production is larger than your degradation, so your arrows are that. Can everyone see what's happening here? You can try to just draw this yourself. But this already tells you enough, all that you need to know about the stability. Why? Because let's say you start from this steady state, and you go slightly on that side. Where are you going to go? You're going to come back to the steady state. If you start slightly on this side, you will come back. So no matter which way you perturb, you will come back. So this one is stable. And this one, because the arrows are pointing outwards, this one is unstable. So now we have a situation where we have two steady states, but only one stable one. So this is not a bistable system. So now we need to make it bistable. So we need to change f of A and try to find another curve that actually cuts it in enough places, in enough ways that at least two of those are stable. And you can already see the rule for stability. The rule is rather straightforward. If you cut from above, if you cut this gamma A line from above, you're going to be stable. If you cut from below, you're going to be unstable. Or more algebraically, if this was the slope of the line at A star, you want this to be less than gamma for stability. That's just an algebraic way of saying it. Okay, so in one dimension, it's rather straightforward. It's easy to just look at a curve and see where you will be stable, where you won't. So we need one more cut that's kind of flat, that cuts from above, right? So the most obvious way to do that, using the kinds of curves we've been looking at, would be to say, all right, I'll cut here, just like this, but then I'll flatten out and cut like this. So here there are 3 cuts, and you can verify that this is stable, this is unstable, and this is stable. Okay? That's one dimension. We've done everything we needed to do in one dimension. In one dimension, it's easy. What I want to do in the rest of the roughly 10 minutes that's available is to now, with all these tools, come back to this and talk about 2 dimensions and show you what you can do in 2 dimensions. Okay, any questions about this? All right, excellent. So let's take this equation. If this is 0, you can rewrite this to say CRO is alpha CRO by gamma CRO divided by 1 plus C1 by KC1 H. Right? All I've done is taken that to that side and just written CRO in terms of C1. This defines a line on this space, this 2-dimensional space. And you can see what that line is. I'll draw it out and you can tell me if it makes sense to you. I'm not drawing it very well, but it'll look like this. This is the line d CRO by dt equals 0. Does that make sense? When C1 is small, then it's basically just alpha CRO by gamma CRO. So it's some value up here, which is alpha CRO divided by gamma CRO. As C1 increases, this quantity goes down as a sigmoid. And this position would be K CRO, the steepness would be given by this, and so on. So I've drawn a particular form which maybe would work for something like H is equal to 2 or something like that. If H were 1, it would look different. So if H were 1, it would look something like that. Okay? Now we need to do the same for the other equation. And the other equation gives you a very similar kind of equation, but it says C1 is some alpha C1 by gamma C1 divided by 1 plus CRO by K CRO. That looks a lot like this, except it's kind of flipped over. So it actually starts here, and this would be alpha C1 by gamma C1, and it would go down like this. So think of the same curve just flipped over. Maybe I should draw it in a different color. So this one is dC1 by dt equals 0. And the same rules apply. Where both the curves cross, that's 0 and that's 0. And therefore, this will be the steady state. So if the curves look like this, there is only one place they can cut. And you will find that very similar things happen as happened in one dimension. You will find, for instance, that in order to get bistability, you need to cut in 3 places. So one way would be to cut like that, would be to have one of the curves like this and the other curve like that. Can you see that I've just flipped it over, the mirror image? This is d crow by dt, and this is the C1 by dt equals 0. And it turns out, so I'll just state the result here. This one will be stable, this one will be unstable, and this will be stable. But now, in 2 dimensions, it's not so easy to just look at the curves and see where is the stability, where is not. In general, in 2D and 3D, you need to know a little more algebraically just how to calculate the stability. So I will give you the mathematical way of doing this so that you at least have the tools to actually calculate and figure out if something is stable or not. So imagine in 2D, any questions about this?
Is there any possibility of neutrally stable?
So yes, so let me tell you in the one-dimensional case. So the thing is, neutrally stable is a particular case that you can get. Neutrally, so for people who don't understand, this is a slight aside, but a neutrally stable system is one where if you are at a fixed point and you move away somewhere, you neither really get pushed away nor do you come back. Okay, does that make sense? You can see that that would require a sort of special conditions. So you can get that kind of situation, but it requires very particular fine-tuning of parameters to get a situation where if you move away, you're neither coming in nor going away. As we saw in the one-dimensional case, in most situations, as soon as your production is not equal to your degradation, you would either get pushed away or pushed back. And that's in general true. There are very particular special cases where you might have a situation. So the case where you would, to answer your question, have a neutrally stable situation would be one where in some sense your production rate goes this and then is equal to gamma A for a while. So in fact, then in some sense you have lots of steady states. And moving from this point to that point neither pushes you away nor pushes you back. But those are very special cases. So we're not going to treat them in any generality. Okay, my time is almost up. So if you have two systems like this, and you have some f which could depend on x, y, and I'm being completely general here, and something else which could depend on x, y. Okay? I've now not written it out in particular form. I'm just using general functions. It really doesn't matter. In this case, suppose this has some fixed point x star, y star. In other words, f of x star, y star is 0, g of x star, y star is 0. In that case, when you're talking about stability, what you want to ask is what happens if you were, so let me define some delta x, which is x minus x star. So suppose your x moves a little away from x star, but not a lot. Let's assume this is quite small. And let's assume the same for y. What happens when you're a little bit away from a fixed point? In that case, what you can do is expand these equations. You can write down equations for delta x by dt. And this will be f of x star plus delta x, y star plus delta y, and similarly here. Sorry, this will be g. These are in general very nonlinear equations, but when this is very small, you can do an expansion around this, and there would be, some of you would know about Taylor expansions. You can do a Taylor expansion around this, and I'll give you in a second what the intuition is. But essentially, you can write this as delta, let me say, df by dx evaluated at x star, y star. All of them will be evaluated. So let me just drop that out. Plus df by dy delta y, and this can be written as dg by dx delta x plus dg by dy at delta y. And all these derivatives are all evaluated at the fixed point. So you can write this as a linear system. And in a linear system, this whole thing can be written very neatly in a matrix form. Delta x, delta y is some matrix which has all these 4 elements. So sum A, B, C, D, delta x, delta y. This matrix is called the Jacobian. And for those of you who know what eigenvalues and eigenvectors are, you want the eigenvalues of these, their real parts, to be less than 0. So that's the rule, that's the mathematical rule. And if the eigenvalues of this matrix are less than 0, you are stable. If they're not, if any one of the eigenvalues has a real part that's larger than 0, you will be unstable. What does that mean? What does that mean intuitively? It obviously has to do, just like in the one-dimensional case, about arrows and where the arrows are going. Anywhere in this space, I can draw an arrow that represents my dc1 by dt and dcrow by dt. The arrow will tell you, if you started at that point, will crow decrease, will c1 decrease? If they both decrease, the arrow will be this way. If one of them, if both increase, let's say, it'll be this way, and so on. So this whole space, you can think intuitively, all these equations essentially just tell you where is the flow going to be. So for instance, it could be like that. This could be one set of arrows which go like this. And the question is, if you are looking very close to a fixed point, one of these steady states, where are the arrows pointing? So here I've kind of drawn out a set of arrows, and you're interested in whether this is stable. You want to look in a little region around this and ask, are all the arrows pointing in? Or are some way places the arrows pointing out? And all this mathematics just tells you that. And what it tells you is there are 2 directions you need to be interested in. Let's take this one. There's a direction here and a direction here you need to be interested in. In this direction, things are coming in, the way I've drawn them. But in this direction, things are going away. And that mathematically would correspond to one eigenvalue being negative and one being positive. This is a stable fixed point. Again, there are 2 directions that are of interest. And here, everywhere the arrows point in. And that corresponds to the eigenvalues being both negative. So generally, whenever eigenvalues are negative, it sort of means that arrows are coming in to the point you're interested in. And when eigenvalues are positive, it means arrows are going out. So all of that is just a simple way of saying, is everything flowing back into this or flowing out in any direction? And you only need to flow out in one direction for something to be unstable. You might be flowing into a steady state in all directions, but there's one direction where you're unstable and you're out. That means the steady state is unstable. So in order for stability, everything has to be flowing into a stable, steady state. So now, sort of both intuitively and with some mathematics, hopefully, you have some of the tools that are required to take equations like this and ask, how many steady states do you have? That just requires plotting these kind of curves, seeing how many times they cross. And the second question is, are they stable? Which essentially requires looking at these arrows. Okay, so let me actually then just stop here. And next time, you will see some of this in action. I will be discussing with Sunil some interesting phenomena in yeast. And he'll tell you about the biology of it, and I'll try to use some of these tools to answer some of the questions he raises as a biologist. Are there any questions about this? We can open up discussions now. Thank you for your patience.